This video will cover subtopic 3.2, chromosomes, and this will be part one of a two-part video. So here are the understandings, application, and skills that we'll cover in part one of this video, as well as a little bit of guidance to help you along. So we'll start with bacterial chromosomes. So bacterial chromosomes have one circular chromosome consisting of all the genes needed for basic life processes. The DNA within these bacterial chromosomes are not associated with proteins and are thus described as naked DNA. There's usually only one copy of each gene since there is only one chromosome. So just remember that you can only have two copies after the DNA has been replicated, but before cell division. Now, there can be additional DNA found within prokaryotes in the form of plasmids. And plasmids are small extra DNA molecules found within those prokaryotic cells. They're usually small, they are going to be circular, and similar to the prokaryotic chromosome, they're going to be naked. So they're not going to be associated with any proteins. Now, they contain very few genes, but they do contain genes that may be useful for the cell, but not necessary for basic life processes. So the prokaryote can exist without having that plasmid. So there's no genes needed for important metabolic processes or cell division found within plasmids. So for example, many plasmids contain genes that would encode for antibiotic resistance. Now, plasmids are not replicated at the same time as chromosomes of that prokaryotic cell or at the same rate. For example, a plasmid may replicate a lot quicker than the bacterial cell chromosome. And so a bacterial cell or a prokaryotic could have many copies of the same plasmid within the cell, but only one copy of its chromosome. So may have many copies per cell, and a plasmid may not be passed to both cells during cell division. So it could be an unequal sharing of these plasmids between cells. So now with reproduction of prokaryotic cells, it can happen through binary fission or a process referred to as conjugation. So plasmids can be transferred from one cell to another through that process of conjugation. You get two cells that join together through a long protein bridge called a pilus, and you can get plasmids of one cell that is transferred to another cell. This allows bacteria to increase their genetic diversity by gaining new DNA, and allows DNA to be transferred between bacterial species as well. So now plasmids have a lot of use in research. So plasmids can be used in research to transfer genes between species, basically from one, we'll say, plant species to another plant species, or even a gene found within a fish can be transferred to a plant. So the way this works is you can take a plasmid, and the plasmid is found within these bacterial cells. Plasmids are circular DNA. You can use an enzyme called a restriction enzyme, and we'll talk more about restriction enzymes later on. But a restriction enzyme will cut open this DNA, this circular DNA, at a particular location, and it will allow a new sequence or a new bit of DNA to be inserted. So that opening can allow the DNA encoding a gene to be inserted within that plasmid. So you can insert your new gene into the plasmid. That will close up the circle. This plasmid can then be transferred into the bacterial cell. And then that bacteria can be used to insert or infect the cell and insert that DNA into that cell of interest. That cell can then be grown in culture, and those cells can eventually grow into an organism, such as a plant. Now, to measure DNA molecules, there is a process that's used called autoradiography. to investigate the shape and even size of DNA molecules. So with the use of these images that were created, it is used to provide strong evidence that will help support a hypothesis. And developments in microscopy have allowed images to be produced of structures that were previously invisible, various cell organelles, for example. In 1960, a researcher by the name of John Cairns used this process of autoradiography to obtain images of a whole DNA molecule from E. coli. So basically, he used it to visualize the chromosome of a prokaryotic cell. From his images that he generated, he concluded that the DNA molecule was a single chromosomal loop. And he also noted where replication forks occur within the DNA. So as the DNA is being copied, you basically get what is called a replication fork, where you get that newly synthesized DNA. So the image provided here is an example of the type of image Cairns would have generated from this process of autoradiography. Basically, the black spots and these black dots are the bacterial chromosome, and then these loops off to the side are the replication forks. So we'll look at in the next slide how these black spots get generated to show the shape of that chromosome. So Cairns produces images of DNA molecules from E. coli through several steps. So he grew cells for 2 generations in a culture that contained a molecule called tritiated thymidine. So remember, thymidine is going to be the T in DNA. It's going to be one of those 4 letters that is incorporated into DNA. And I know this says titrated, but it should say tritiated. Basically, it's a thymidine containing an atom of tritium. And what is tritium? So tritium is going to be an isotope of hydrogen. So remember that hydrogen is an atom that contains 1 proton and 0 neutrons. Once you start to add in neutrons into the nucleus of hydrogen, you can start to change the mass of hydrogen. And we give these different isotopes of hydrogens different names. So once you add in a neutron into the nucleus, you now get a hydrogen atom that's called deuterium. If you add in 2 neutrons, and that's what the 3 is going to represent, and you still only have 1 proton. Hydrogen is always 1 proton. But now you have 2 neutrons. And this is that element, or the isotope tritium. Tritium is radioactive. And so incorporating this radioactive element into thymidine means that as the DNA is replicated and new DNA is synthesized, the thymidine that gets incorporated into the DNA is going to contain this radioactive DNA. And now, yes, it is harmful for the cell, but they can grow in it for a little bit. So he grew 2 generations of E. coli in this culture containing this radioactive thymidine. And after 2 generations, once these cells now had some radioactive DNA, the cells were then digested and the cells were burst to release their DNA onto a dialysis membrane. So the DNA was released onto a dialysis membrane. This radioactive DNA is giving off radiation, and that radiation can actually be detected by placing a film on top of the membrane. And this is like camera film that, if exposed to light, it'll turn black, or exposed to radiation will turn black. So this film was placed on the surface of the membrane to allow the tritium in the DNA to decay and emit high energy electrons that react with the film. Where the tritium decayed, one will see a dark grain on the film, and these indicate this indicates the position of the DNA. So what you're seeing on this image here with these little black spots, these are areas where thymidine exists within the DNA and where radiation was given off and made contact with the film. And so what this told Cairns is that the DNA is found in a loop. So, as I mentioned, it showed that the chromosome in E. coli was a long singular molecule that was about 1100 micrometers in length. This technique was then used to determine images of eukaryotic chromosomes. So, the fruit fly, the Drosophila melanogaster, was one of the first images, and its chromosome was imaged to be about and determined to be about 12,000 micrometers long, so significantly larger than that bacterial chromosome. Moving on to eukaryotic chromosomes. So eukaryotic chromosomes are composed of both DNA and protein, and we're talking about protein in the form of histones. And this diagram here shows how DNA is condensed and folded, and structured at different levels. So, in the nucleus, you have that chromatin structure, which is basically coiled DNA around the histones, but it's basically an uncondensed version of a chromosome. During DNA replication, this chromosome will condense and supercoil into a chromosome. For those proteins, those histones, you can think of them as beads on a string. So histones are globular in shape and are generally wider than DNA. Now, there are many histone molecules in a chromosome with the DNA molecule wound around them. Adjacent histones in the chromosome are separated by short stretches of naked DNA. So you have your DNA that gets wound around the histone, and then in between the two histones, there's going to be a stretch of DNA that is not associated with any protein, and that will be naked DNA. And usually that naked DNA is going to be DNA that is going to be actively transcribed or actively used by the cell. So with those histones now, eukaryotic chromosomes are linear, and they're basically going to be made up of DNA and histone proteins. Those histones, as mentioned before, are globular shaped with DNA wrapped around them. So you get DNA wrapped around 8 histone proteins. That's referred to as a nucleosome. So you have basically 4 histone proteins and 2 of them in a nucleosome, and then you get the DNA that wraps around those core of 8 histone proteins. There is another histone protein called H1 that is attached to the outside of the DNA strand. So outside, kind of clamping that DNA onto those histone proteins, is that H1 histone protein that is not circular. And as discussed before, DNA is negatively charged, and because of this negative charge, it is going to be attracted to the positive charge on the histone proteins. And so histone proteins generally have a lot of positively charged amino acids on them, and so this creates that positive attraction between them. So now there are differences between chromosomes within eukaryotic cells. Generally, though, eukaryotic chromosomes are too narrow to be visible with a light microscope during interphase. So if you think about the cells that we've stained before, when you looked at the nucleus, it showed up as a brown stain, and that's basically all of the chromatin being stained. However, during mitosis and meiosis, the chromosomes will become condensed by supercoiling and will become visible if stained. So you can start to see individual chromosomes during mitosis. So this picture shows those individual chromosomes that have now condensed by supercoiling. And these are the 46 human chromosomes. When chromosomes are viewed during mitosis, there are many different types of chromosomes can be seen. These different types of chromosomes will be different in length and the position of the centromere. And there are at least two different types of chromosomes in every eukaryote. So every human has 23 different types of chromosomes. They have two of every type of chromosome. Therefore, in total, they have 46 chromosomes. Now recall that every gene in a eukaryote will occupy a specific position on one type of chromosome, referred to as a locus. Each chromosome type therefore carries a specific sequence of genes. Having genes in a standard sequence along a type of chromosome allows parts of chromosomes to be swapped during meiosis, and we'll get into more detail about how that occurs. Because we have two of every type of chromosome, those two types of chromosomes, or those two pairs of chromosomes, are referred to as homologous chromosomes. So homologous chromosomes can be defined as two chromosomes that have the same sequence of genes. Now, they are not going to be identical because they vary in their alleles. So remember, alleles are the variations in those genes. So between those two chromosomes, there can be different alleles that exist. They'll have the same sequence of genes, but those genes can vary based on those alleles. So where do those two chromosomes come from? Remember that one chromosome will come from dad, one chromosome will come from mom. And because these two chromosomes will have the same sequence of genes and will have the same position of centromere, they will be referred to as homologous pairs of chromosomes. And what this diagram indicates is, or basically what the diagram shows, is here we have unreplicated DNA. And then when the DNA replicates, we get this familiar looking X pattern of our chromosomes. So here we have, one full set of genes, and then here is our duplicated set of genes. Remember, the replicated DNA is made up of sister chromatids. So once we have all those chromosomes figured out in terms of length and size, we can now get down to comparing genome size between different organisms. So the genomes of living organisms will vary. Viruses have the smallest genomes, but in IB they're not regarded as living. So IB does not consider viruses to be living things. So therefore, prokaryotes will have the smallest genomes. The genome of a eukaryote now depends on the size and number of chromosomes. It is correlated with complexity, size and number of chromosomes, but not proportion. The proportion of DNA that acts as functional genes is variable, and also the amount of duplication varies. So when we compare genome size between different species, just for the sake of comparison here, we have a virus. This is referred to as a phage virus, which infects bacterial cells, specifically E. coli. And they have a very small number of, or their genome is very small when talking about base pairs, compared to E. coli, where we now get into millions of base pairs. Compared that to fruit fly, where now suddenly we're into hundreds of millions of base pairs, and compared to humans, there are many thousands of millions of base pairs. But you can get into plant species where the size of their genome is significantly larger than ours. So that is it for this video for part 1 of 3.2 chromosomes.